Okay, so it is the continuation of rRNA. Okay, we're talking about ribosomes. rRNA makes up the ribosomes. So this R stands for ribosome RNA, and this is produced inside the nucleolus, which I shall discuss a little later. So rRNA, where it is produced, the first initially produced rRNAs are called pre-RNAs. It needs to be spliced, or it needs to undergo splicing. What is splicing? It is the removal of the introns, all the non-coded regions, or regions which does not contain codes for protein synthesis or the formation of RNA in this case. And when it is spliced, it forms one large structure, one large piece, and another small piece. So this is very important. When it is broken apart, when it's spliced up, it turns to form one large part and a small part. So this large part eventually forms the large ribosomal subunit. So look at the structure of ribosome. It contains two subunits, a large one and a small one. So the large ribosomal subunit is formed by the large spliced portions, and the small pieces form the small ribosomal subunit. I've started with the small ribosomal subunit because it has a small role to play, and its role or its function is to attach to the mRNA cap. So here is an mRNA, a mature mRNA containing a cap and a tail. So this is its 3' end, and this is its 5' end. So an RNA actually attaches here, and then it begins to move down towards the 3' end, and it begins to read these codes written in combinations of 3's. These are codons. Okay? So that's how this mRNA, the ribosome works. It fixes itself on the cap and moves towards the 3' end of an mRNA. A large ribosomal subunit. So that was the work of the small ribosomal subunit. Fix itself on the cap of the mRNA and begin to move towards the 3' end. The large ribosomal subunit contains 3 binding sites for the tRNA. So here is a tRNA, and it has, now this tRNA is also known as amino acylated tRNA. What it means is, it's a tRNA which is fixed with an amino acid. So a tRNA has, on its 3' end, a tRNA has a binding site for an amino acid. Okay? So the type of amino acid that is supposed to be bound here at the 3' end is dependent on the anticodon, or the codes that's written here. These 3 combination. This 3 codon determines the type of amino acid that needs to be fixed on the 3' end. So there are around, let's just say around 40 to 60 different types of tRNA. Just floating in the cytoplasm. There are 40 different types because of the codons here. Different types of codons, different types of amino acids attached to the tRNA. So the number of different types of tRNA with different types of amino acids is around 40 to 60. Let's come back to the large ribosomal subunit again. It contains 3 binding sites for the tRNA. So this tRNA needs to combine with the large subunit of the ribosome. It contains 3 sites. One is the A site, P site, and the E site. A site is amino acyl. This A stands for amino acyl. It binds to the amino acylated tRNA. So it is a tRNA which is attached to an amino acid. acid. tRNA plus an amino acid is amino acylated tRNA. Okay? This simply means a tRNA. I'm just going to draw one tRNA here with a particular type of amino acid on its 3' end. So here is its anticodon. This is the anticodon. This anticodon has to correspond to the codon written on the mRNA, just like this. Here is the mRNA, and here is the tRNA with its anticodon here, which corresponds to this codon of the mRNA. So this is the anticodon. So this actually pulls one type of tRNA. This actually pulls one type of tRNA, okay, which corresponds to this code. And when it does, it also pulls another amino acid which is fixed on the tRNA. So here is the tRNA. It comes to the first binding site and then it moves to the second binding site where there is a channel that I've drawn here. There's a hole here that runs through the large ribosomal subunit. From here, the amino acids come out. So these are the different amino acids that have already been, pumped into that canal of that passage. So as this tRNA comes in here, and the mRNA moves to this side, it gives out its amino acid that it was carrying, and that amino acid joins to the peptide chain. The polypeptide chain that has been formed is being added with another amino acid. Okay? So now this tRNA is without its amino acid. It goes here to the E binding site. E binding site is called the exit. It holds the deacylated. Deacylated meaning a tRNA which has given off its AA. It does not have an AA. So this is a deacylated tRNA. So here, the deacylated tRNA is held on, okay, for a little while in the E site and then let to move away. So this comes out of the ribosome and it goes in search of its amino acid that needs to be fixed there. According to the anticodon or the codes that's written here, it needs to pull a new amino acid. So this is how it works. The ribosome comprises of two parts, the large ribosomal subunit and the small ribosomal subunit. Small ribosomal subunit actually fixes itself on the cap of the mRNA and begins to move towards its tail. And the large ribosomal subunit contains three binding sites. They are A site, P site, and E site. On the A site, it grabs on or it latches on to an amino acylated tRNA. What is an amino acylated tRNA? It is a tRNA which contains an amino acid that is dependent on the codes that's written on its anticodon site. Okay? So anticodon and the codon on the mRNA, they correspond, they attract each other. And when this codon passes through the A site, it pulls its corresponding, the correct type of tRNA into itself so that it goes into the next binding site known as the P site, where it gives away its amino acid and that amino acid joins the polypeptide chain to form what's known as the protein and the protein synthesis. And I've drawn this structure here where again we have this pentyl sugar with this is the nitrogenous base, this is the pentyl sugar, and here you have a phosphate group bound to the 5' end. So this is the 5' and the 3' end. So if I have to draw a very simple structure of this nucleotide, I'm going to draw it like this. So this is the 5' end and this is the 3' end. So this is very important in the next stage of what you call, not next stage, but during the process of what you call transcription. Okay, there you will find this point very important. So we still have